Let's now see the structure of the sarcomere. It's a high yield topic. Lots of questions are asked on this topic. Well, inside the muscle, there are muscle fibers, and inside each fiber, this structure will be seen. Thin and thick filaments within each muscle fiber. So, what's a sarcomere? At regular distances, there are zigzag lines called as Z lines. Arising on either side of the Z line, there are thin filaments. And among those thin filaments, there are thick filaments. Interspersed among the thin filaments will be the thick filaments. That's thin filaments arising on either side of the Z line. Among these thin filaments, then there are thick filaments. That is the organization of thin and thick filaments. There was an MCQ related to this ratio of thin to thick filaments. What is the ratio of, or rather, each thin filament is surrounded by how many thick filaments? Each thin is surrounded by how many thick filaments? And the options were 2, 3, 4, 6. Well, the answer is 3. Each thin filament is surrounded by 3 thick filaments. Let's see how. First of all, let us discuss the length of the sarcomere. That's the first issue. Distance between two successive Z lines is the sarcomere. It is the structural and functional unit of the muscle. Structural unit. Functional unit is rather the motor unit. Is the functional unit. Let's see that little later. Okay. There are Z lines at regular distances. So there will be Z line here, and these regular distances, the distance between two successive Z lines is the sarcomere. Now, the normal resting length of the sarcomere is 2 microns, and this is called as L initial or L resting. Length resting or length initial is 2 microns. Now, there is Starling's law. The Starling's law for skeletal muscle contraction or a muscle contraction, it says greater the initial length, stronger is the contraction. Initial length of the muscle fiber, and therefore it would be initial length of the sarcomere as well. So, greater the initial length means when the muscle is beginning to contract. What's the length? Greater that length, stronger is the contraction. But within physiological limits only, this length ratio is applicable. So, you increase the length of the muscle, increase the length of the muscle fiber, and increase the length of the sarcomere. 2.1 microns length, stronger contraction, Starling's law. 2.2 microns, even stronger contraction. 2.2 microns is said to be the length optimum, LO. This will produce strongest possible muscle contraction, because at this length, the actin-myosin interdigitation is said to be optimum. Initial length of the sarcomere, if you stretch the fiber, it increases, stretch it further, length increases. It's a good interdigitation between actin and myosin. It will produce strongest possible muscle contraction. So that's length optimum. Now, you stretch the fiber further, increase the sarcomere length further, the contraction will become weak after this, because the actin-myosin interdigitation will be lost beyond this length. So stretch the fiber and actin-myosin interdigitation will be lost. The contraction strength will become weaker, and about 3.7 microns is called as L max, length maximum. From this starting length, the contraction may not be strong at all. In fact, it will be hardly any contraction, zero contraction from this starting length. That's the length of the sarcomere and its relation with the contraction. Length tension relationship, it's called, and the basis is Starling's law. Second issue is a ratio of thin and thick filaments. Now, this is a two-dimensional view, but in reality, sarcomere is a three-dimensional structure. It's inside the muscle fiber. So, if you see the transverse section across the Z line, the cross-sectional view of the Z line, it would be a hexagon. The Z line is actually a hexagon in the cross-sectional view. Arising from each of its angles, there is one thin filament arising from each angle of the Z line, and among these thin filaments, the thick filaments are interspersed. So that's how it will appear on cross-sectional view. It means each thick filament will be surrounded by 6 thin filaments like this, and each of the thin filaments will be surrounded by 3 thick filaments. If I consider this thin filament, it will be surrounded by 3 thick filaments like this. So each thin surrounded by 3 thick filaments, and therefore in the skeletal muscle the ratio of thin to thick is 2:1. It's different for other muscle types, but for skeletal muscle the ratio is 2:1, and this is the interdigitation between of the thin and thick filaments. It's like this. That was the thin and thick filaments and their relation to each other. Second point. Third point is, let us talk about the muscle proteins. 2 muscle proteins. One is titin, the other is dystrophin. One MCQ was asked in some of the recent exams was that largest known human protein is, and the options are dystrophin, titin, myosin or laminin. The answer is titin. Let's talk about the titin. Well, titin is anchored to the Z line and it enters the thick filament. So that's one. Titin, it's the largest known human protein, almost 41,000 amino acids and 3 million molecular weight. It's the largest known human protein. It is anchored to the Z line and then it enters the thick filament. What is the function of the titin? Alignment of the thick filament in the sarcomere. That's the function of titin. Alignment of the thick filament in the sarcomere. That's the job of titin. And the genetic defect of titin, genetic defect in the gene that codes titin, results in a defect called as limb girdle muscular dystrophy. It's a titinopathy. Limb girdle muscular dystrophy is a titinopathy from the genetic defect of titin. That's one muscle protein. Another muscle protein which should be known is the dystrophin. What's the function of dystrophin? See, dystrophin is linked to the thin filament. So starting from the thin filament, and it reaches the sarcolemma. The muscle fiber membrane is called as sarcolemma. In the sarcolemma, there are glycoproteins. So this is the so-called dystrophin glycoprotein complex. This is dystrophin. It starts from the thin filament and it goes to the sarcolemma. In the sarcolemma, there are glycoproteins. In all, there are 6 glycoprotein subunits. What are they? Dystrophin is directly linked to the beta-dystroglycan, which in turn is associated with alpha-dystroglycan. So 2 subunits of dystroglycans, and these dystroglycans are in turn associated with 4 subunits of sarcoglycans. Alpha, beta, gamma, delta sarcoglycans. 2 subunits of dystroglycans, 4 subunits of sarcoglycans. Dystrophin therefore is associated with 6 subunits in the sarcolemma. Alpha dystroglycan in turn is linked to an ECM protein called as laminin. Laminin is the extracellular matrix protein. And thus, what is the function of dystrophin? You see, thin and thick filament interaction will generate tension in the muscle. Contraction will happen. And the tension generated by thin and thick filament interaction has to be transferred to the outside of the muscle fiber membrane and then to the tendon. That's the job of dystrophin. Tension generated by actin-myosin interaction is transferred to the exterior of the muscle fiber membrane and thence toward the tendon of the muscle. That's the role of dystrophin. And genetic defect of dystrophin, or the gene that codes for it, the defective gene, will result in Duchenne's muscular dystrophy, DMD, Duchenne's muscular dystrophy. That's the dystrophin, another muscle protein. Let's now talk about the various bands and zones in the sarcomere. Let's see various bands or zones in the sarcomere. This one question has been repeatedly asked. What are the various changes in sarcomere with muscle contraction? Let us first see the bands or zones in the sarcomere. First thing, on either side of the Z line, some distance, there is only thin filament. So this is called as I band, isotropic to polarized light, therefore I band. If you pass the polarized light through this area, since there are only thin filaments, their refractile property will be similar, and it's called as I band, isotropic to polarized light. Remaining part of the sarcomere would be called as A band, anisotropic to polarized light, because this band contains thin as well as thick filaments which have a different properties to the polarized light. It's called as anisotropic or A band. If you notice, one sarcomere is equal to one full A band and 2 half I bands. This half I band here and this half I band. That's the sarcomere. And we saw sarcotubular system in the skeletal muscle. There were 2 T tubules per sarcomere, which I mentioned. They are at the junction of I bands with A bands. So here at the junction there would be T tubules in the case of a skeletal muscle. Whereas in the cardiac muscle there is only 1 T tubule per sarcomere. It's lined along the Z lines. So this will be the T tubules in the cardiac muscle, like that. Right. At the very center of the A band, there is H zone. H zone. The H zone will have only thick filaments, because ends of the thin filaments, they have ended just short of the center, and therefore there is this gap which is called as H zone. And at the very center of the H zone, this is at the very center of the sarcomere, there is a dark line called as M line. There is a protein called as myomycin over here, and it's called as M line, the dark line. There is one more zone or band on either side of the M line. There are light areas. If you see at the very center of the H zone, there is M line. If you pass light through this area, there is dark line called as M line. Now either side of the M line, some distance, there are light areas on either side. Dark line at the center, and then immediately adjacent would be the light areas. So the dark M line plus the adjoining light areas, this is called as pseudo H zone. M line plus the adjoining light areas on either side. This is the so-called pseudo H zones. The question which is often asked is, what are the changes in sarcomere with muscle contraction and shortening? Which band or zone becomes prominent with muscle contraction? That MCQ was asked recently, and the options were I band, H zone, M line, or pseudo H zone. Which band or zone or line becomes prominent? And the answer was M line. Let's see how. You see, when the muscle contracts, entire fiber will shorten towards the center of the fiber. There will be shortening of all sarcomeres toward the center of the fiber. So the sarcomere will shorten. The Z lines are pulled inward. For all the sarcomeres are shortening. Therefore, sarcomere length will shorten. Thin filaments are pulled, their ends are pulled towards the center of the sarcomere. That's shortening of the muscle. Entire fiber is going to shorten. So all sarcomeres are going to shorten. And these are the changes which contraction and shortening. First change, I band shortens, because as you can see, the shortening of the sarcomeres, ends of the thick filaments will come closer near the Z lines, and I band will shorten. Second, it's not a change, but we can say A band remains unchanged. A band remains unchanged in its width, because A band width is the width of the thick filament, and there is no change in the width of the thick filament. Thick filament is not going to shorten. It will remain so, and therefore A band remains unchanged in its width. Third thing, H zone disappears. H zone will disappear. As you can see that thin filaments have begun to overlap just at the center of the sarcomere. So that gap which was there will disappear. And the next change is M line becomes prominent. M line now will become dark and prominent. As you can see, the overlapping thin filaments, they have been added in this zone now. So that dark M line at the center will become dark and wide, and therefore we will say M line becomes prominent. that central dark line. In fact, two new zones will be added after contraction. The two new zones would be at 1.5 micron length of the sarcomere length of the sarcomere length. Now we are talking of the contracted length. Earlier we discussed the initial length of the sarcomere when the muscle was resting or muscle was about to start its contraction. Now we are talking about the contracted length of the sarcomere. At 1.5 micron length of the sarcomere, a new band will appear which is called as CM band. C stands for contraction and M line now is converted to M band. Next, with very severe contraction, 1.25 micron length, another band will appear in the sarcomere which is called as CZ band. Contraction, Z band. The darkness around the Z line, because now there is so much shortening that the ends of the thick filaments will be very close to the Z lines, and therefore another dark area created around the Z line called as CZ band. Contraction, Z band. These are the bands in the sarcomere, skeletal muscle sarcomere, and these are the changes that happen with muscle contraction.